don't rely only on the slides, okay, which I was discussing the points. May I also missed some points sometimes. Because the syllabus for SRF is very large, like it's unlimited. Every day the new concepts are coming and every day the new knowledge is coming up in every aspects. For example, you take in markers, maybe some new marker systems are exploited very in a large scale that may be asked as a question. There are some schemes of marker selection, some different advancement in genomic selection, or some advancement in cytogenetics. Every day it's a growing science, and the syllabus it's not like a rigid like for JRF. It's a very like dynamic. So that always keep reading some advanced like not advanced topics, some important journals, for example, Nature Genetics and crop breeding, tag, and some keep on reading some reviews on different topics like behind the Jivas, there is some papers and some behind like paradigm shifts in plant breeding. There are several like today I was listening to a lecture on conferences going on hybrid technology. There was discussion about um, they're all renowned scientists of plant breeding. They were there. There was a discussion about the five paradigm shifts in the plant breeding. We're discussing about how apomictic can help, how apomix is like apomictic one-line breeding, and another thing is uh, reducing the like reducing the number of phenotypes and increasing the genotyping of individuals means prediction based breeding like predictive based breeding. So many things coming up, and there are so many things like rapid advancement generation techniques. So many things are coming up. Should be in touch with all the topics and keep on listening some lectures and don't miss any invited lectures, especially from renowned speakers. Today there was a speaker called as Suganeshwaran. He's actually discovered a synthetic apomictic lines. Since natural apomicsis is available, he discovered synthetic apomictic lines. The gene which has responsibilities, baby. Now, come on. No one is there. Please be with the topics. Be sure with the topics. And today I was going to discuss about very important topic called as reverse breeding. Because the paper, I think only one paper is there, published in Plant Biotechnology Journal. It's a very renowned journal. This paper, please read this paper, everyone. Okay, published in 2009 only. It's actually a review article. There is another paper also. Okay? It is published in same journal, Plant Biotechnology Journal. You just read, and this review is very very important. Rob review. Reverse breeding, a novel breeding approach based on engineered meiosis. How many of you know about reverse breeding? Can anyone tell what is reverse breeding? Can anybody tell what is reverse breeding? If any have an idea what is reverse breeding,
Development of homozygous parents from F1 generation.
exactly. Great. Okay, my screen is visible, like my PPT, my slides are visible.
Yes, sir.
It's actually it's published in the review is published in Plant Biotechnology Journal. So it's a NAS rating about 20, I think. It's a very important journal and very good journal also. And we're splitting. It's a just development of homozygous lines from the heterozygous parents. Okay? If you have an heterozygotes, you develop homozygotes. So we can able to constitute and we can use and again able to cross this and we can able to constitute a heterozygote. Why we need a reverse breeding technique? We will discuss. Okay? For example, what is the nature of hybrid? How we need to produce an hybrid? How hybrid seed production will take place?
After our two parents, exactly to
How?
produce nitrogen.
Hello?
If it is if it is a cross-pollinating crop, we should develop hybrids and then
Yeah,
we should breed them. If it is a
yeah.
self-pollinate crop, we can directly go for any sterility systems and breed them.
Where it is easy, either in cross-pollinated crops, self-pollinated crops. That's another topic, another discussion. Okay?
Cross-pollinated crops there.
For hybrid, we need how hybrids is produced every year. You need to cross and inbreds. Okay? You need to cross and inbred either any male sterility systems or whatever it may be, but you have to cross male and female. Why the male sterility system is used? Because if it is a self to emasculate genetically the male gametes, we are using the all male sterility systems. Okay, that's not the issue. Okay, but whatever it may be, you have to cross. We need a parents, inbred parents. Then you are crossing F1, and this process is continuous because we can't self F1. There will be segregation. That's why for every seed production we need. That's why whenever you see any hybrid, they must be provide two parents and one hybrid seed. Okay, their parents along with hybrid seed is applied, and every time you need to cross parent one parent two, then create hybrid. This is the only. But in system in nature, there are several landmark hybrids for there. But during this one, their parents is lost. During this one, their parents is lost because some are naturally heterozygous. So like other crops, but some are like hybrids are there. But unfortunately, you don't have parents for that. What we can do? You can't self the his hybrid. If you self it, what happens? The etrotic potential of this hybrid will going to lost. We need a parents so that we can we need a homozygous parent, like completely homozygous parents inbreds, so that we can cross and we can get an F1. So that it will be highly heterotic in nature. To get the parents, how we can develop the parents from a child? It's possible. It is possible in case of plants. Okay. For example, to have a renowned hybrid, but you lost his parents. Don't know who is parent of this renowned hybrid. You can't self and you can't generate the hybrid like this. May your heterotic will be going to loss. Like heterosis will going to be lost. Heterotic potential will going to loss. In that case, what we can do? We have to reconstitute the parents. To reconstitution of the homozygous parents from the present hybrid. It is called as reverse breeding. We are going back to the parents. Okay? Through how we can do? Through engineered meiosis. How exactly the process will take place? We will discuss. What is reverse breeding? Reverse breeding allows the fixation of a complex heterozygous genomes by constructing a complementary homozygous line. Means from F1, we are creating a complementary parent 1 and parent 2. So that if you cross that, we can able to reconstruct this F1. Then, how we can achieve this? By suppressing the meiotic crossovers and generating a double haploid lines with a non-recombinant chromosomes. For example, if you self it, what will happen? There will be recombination. Means there will be exchange of chromosomes so that the integrity of the chromosome will going to last. The original native nature of the chromosome will going to last. They will recombine and they will segregate in a n number of different ways based on the number of genes they were segregating. Okay, but you need to avoid that. You just stop the recombination so that the chromosomes will remain intact. They will not going to recombine. They will remain intact so that we can able to do something like double haploid technique so that we can able to reconstitute the complementary parents. Okay, this is what in nutshell uh, reverse breeding means. Okay, whenever this somebody asks you reverse breeding, should able to deliver this definition. Okay, then where it can use fixation of some uncharacterized germplasm with desirable traits. For example, it is in heterozygous condition. Whenever it's selfing it happen, it will segregate. It, it will segregate and it will lot some desirable character. So that you want to fix. How we can fix? We can develop their parents in homozygous condition so that fix these parents. Whenever you need, you just cross them, you will get that drug. But when you keep on selfing, what will happen? The chances of getting identity, the segregation will be more. The getting more better recombination will be very, very less. Those who know about the principle of heterosis, it will be very easy to understand. Okay. Then you can do the breeding at chromosome, individual chromosome level that we will discuss in these slides. Okay, first there will be heterogeneous population and superior heterozygote, but we don't know their parents. We include we're doing the engineering meiosis. This is called as reverse breeding. Then we can able to develop the parental lines P1 and P2, so that whenever you cross, you can able to generate this superior heterozygous plant. Okay, this is called as reverse breeding. Understood what is reverse breeding? Hello, anyone is there? Hello, Venkat, understood? Somajit,
Yes, sir. Yes, sir. Until now, I understood.
yeah. Next, how exactly it will happen? We will see. For example, you take there is a F1 like this. You lost these parents. Okay. If you do a segregate, if you there will be if you do F1, there will be selfie. There will be an F2. If you do reverse breeding, okay. If you do a reverse breeding at F2, what will happen? There it will stop the recombination between them. So it will stop the recombination between them. You think these are the gametes? Here from one gamete, these are the gametes. If you stop recombination, you can get an intact. You can get an intact gamete. See, there is no recombination between. If you're doing, you can stop recombination at two levels, either in F2 or recombination. You can stop recombination either in F1. Two ways you can able to do. For example, first, by the time purpose, you can see here this is an F1. If you stop recombination here only, it will not going to recombine. So you are getting an intact chromosome. See, intact chromosomes. If it is recombines, what will happen? Like this, it will come. There is some portion will be this color, some portion will be this color, some portion will be this color, some portion will be this color. Means here we are not stopping recombination here. In case of sorry here in F1 we are allowing the recombination so that F2 segregance is produced. But F2 you stop the recombination so that next progenies will have the intact chromosome so that we can able to reconstitute the good hybrid good this we can reconstitute this one and we can able to develop the complementing parents of this and see this one and this one, it is exactly same. You can observe this pattern of segregation here and here. Okay, this heterozygote, if you stop the recombination between them, you can able to get the intact individual like this. That is the crux of the this technique. Means you have to stop the recombination. When you are stop your recombination, there is no exchange of the chromosomes and there is the stoppage of segregation of your genes and recombination. Then what you will get? You are getting an intact chromosome so that you can isolate them. You just multiply. By chromosome doubling, you will getting the homozygous of every chromosome so that whenever you cross with normal, you can able to get the complementing this one. This is what you are just stopping the recombination whenever you want. Either you can stop the recombination at F1, either you can stop the recombination at F2. Both can be possible. Even there's some advanced question may I going to ask. For example, you are using a reverse breeding. At what level you are stopping the recombination, either in F1 or F2. Both the answer will be right. You should answer in. You're able to should able to answer. Even the questions will be twisted. Understood? How we can stop by stopping the recombination? How we can get the intact parents? Understood or not? Okay. I hope you understood.
Sir,
Then
can we
how
get,
you?
excuse me, sir.
Yeah. Yeah.
Sir, can we get the exact parents genome in, if we stop the recombination in the first size, sir, first one
Yeah.
in F2?
Here, no. Here, this one is your hybrid, starting hybrid. See, you are getting the complement parents. Whenever you cross this complement parents, you can able to reconstitute this F1. Okay, reverse breeding is not only the objective of the complementary parents. Whenever you cross the complementary parents, you should able to create the hybrid like earlier. See, this one and this one you can see. The hybrids are this starting hybrid and this hybrid, reconstituted hybrid is exactly looking same. Means the heterosis will remain same. That will be the major object. Okay?
Sir, one doubt.
Yeah.
So how come we know that exactly those two lines after segregation, but without no crossovers, can give this
Yeah, here.
appropriate hybrid, sir?
Yeah, that's
How
where
we
we
are
need to
finding
use
those
some
exact two lines?
it is you have to use cytological techniques. Fish, dish, that's the very difficult actually. Getting like this, it is. I will explain in coming slides how. See, in a simple term, okay. Whenever this very limited here, only four is there. Only four chromosomes are there. Only limited. You are showing in the figure only limited recombination, so that it easily you can okay this one. Okay, but it is a trial and error based on cytological techniques, chromosome painting, or a fish or a dish. There are some genome analysis techniques. We can identify this chromosomes. Okay, these were like this. Okay, if you cross them, maybe you are going to this particular hybrid. Like that, you have to identify by some several cytological techniques. Okay,
Yes, sir.
it's very difficult actually. Getting exactly these two uh, the gametes, it's very difficult. Whenever it is chromosome more than 12, reverse breeding can't be used. That's why reverse breeding still in a review. Okay, nobody is using. The crops which having chromosome number about 12, we can't use reverse breeding. It is very difficult to get this hybrid, like these combinations. Understood? Theoretically, you just think, okay?
Yes.
Then, see, we can able to this one generate. Then, how we can suppress the recombination? There are several ways. I can how we can suppress the there are several genes like mutants we can use. For example, major gene is pair 2 and ASI1 is helps to meiotic crossing over in the meiosis cell division cycle. But you just mutate them, there will be stoppage of recombination. This is characterized in the rub depths. Okay. Then other methods like RNA, we can use RNA silencing techniques, RNA interference techniques, so that you just suppress the genes which are responsible for recombination by RNA. Either you can use dominant negative mutations so that we can able to create the dominant negative mutation so that it can't able to express. Then either you can use virus induced gene silencing. We can suppress the genes which are responsible for recombination. Anything. These are the tools they are used to knock out or knock in some gene expression. You can use any other technique. You can use nowadays CRISPR also. Just you have to suppress, knock out the genes which are responsible for recombination. Either you overexpress the genes which will suppress the recombination. That also true. Maybe the question will ask in the two types. You can knock out the genes which are responsible for recombination. Either you overexpress the genes which are suppressing the recombination. In two ways we can able to stop the recombination. Okay. Either use you can use if you want. These are all transgenics. Okay. You have to introduce some genes so that it will make. But you can use non-transgenic based also. Either use graph based techniques. Either you can use some CRISPR methods. It's a non-transgenic method. There is many things, but ultimate aim is to stop recombination. There is another easy technique called as mirror. It is asked several times in the exam, SRF exam. 4 years, the questions on reverse bleeding is repeated. If you apply the mirror, it's a chemical which will prevent your meiotic recombination. So that way, there are several ways. Either you can create the mutants, either through any river genetic tools, we can able to knock out or knock in the expressions of the particular genes which are responsible for recombination. Either you can apply the mirin so that this chemical will suppress the recombination. In this ways, we can able to stop the meiotic crossovers. Then, um, but what will happen? Okay, we are suppressing the recombinations. What will going to happen? If there is a no recombination will happen, then definitely your meiotic cycle will disrupted. Means if meiosis will happen, you will get a balanced gap. So that proper distortion of your chromosomes to the different poles, so that you will get the gametes which are the segregation of chromosome will be proper and will get viable gametes, else fertile gametes. But if you are doing something to the meiosis, then definitely there will be a distortion of chromosomes. Maybe you will get some sterile or unbalanced chromosomes. Because cell meiosis is an important process where your proper segregation of chromosomes is assured to the gametes. If there is a meiosis, there will be a proper halving, like a proper separation of the chromosome so that you will get an applied gametes. But if the meiosis is disrupted, definitely it will have a undesirable consequence on the fertility of the your pollen or any gamete. Okay, oval or pollen. Then how we can eliminate? Even though if there is a non-crossovers, the probability of getting a balanced spores having all the chromosomes, it will be viable and fertile. It is just 1 by 2 power x. 1 by 2 power x, where x is your number of chromosomes. Means, for example, you have chromosomes 5, like Arabidopsis. What is the probability? 1 by 32. Means, if you're producing 32 gametes, there is a chance of getting only one balanced gametes that is fertile. If your chromosome number is 6, 1 by 2, 6. What will happen? 1 by 2 power 6, how much? 1 by 2 power 6, how much? 2 power 6 what? I think 256, huh?
64.
Yeah, 64. Means out of
No,
64, you are able to get only one viable gamete. If it is more than 12, 2 power 12, how much? 2 power 12? 64 into 64. Several thousands, okay? See, for example,
4096.
yeah, 2086.
4096.
Yeah, 4086 means out of 4086, the chance of getting the balanced spore which is fertile, it is only one. And you have to screen all 4000 like this. The 4000 possibility of this type, that too from 12 chromosomes, it is very, very difficult. That's why nobody is using the reverse breeding. It's a very like, it's like still in a dreams, okay? For many crops, okay, okay, up to okay, some Putinea, Arabidopsis, where your chromosome number is less, they're also very difficult because you have to cytologically to analyzing cytology just PUC. I think you are like in 12 plus. Just observing meiosis, it's difficult. Okay, you have to fix the cell, then you have to observe the chromosomes, then okay, proper segregant. It's a very difficult and tedious task. That's why it is in progress. Okay, but there is a one technique is available so that we can able to get the parents from the F1. You understood what is the limitation of this technique?
Yes, sir.
Definitely, maybe this year they're going to ask. I think if possibility is high. Okay, any doubts? We're asking some doubt.
No.
Yeah. Next, as I told, whenever the higher number of chromosomes, it's very difficult. Okay. Then, what is the advantages of reverse breeding? Why we need to do? Okay, why this? Why we need to do this much effort? Means first one is as I said, we can able to reconstruction of heterozygous germplasm so that by creating complementary high parents by using engineered meiosis, we can able to cross that complementary parents. We can able to reconstruct the heterozygous germplasm. Means, for example, where the crops which having extensive breeding lines. For example, some crops are they don't have some breeding lines, inbred stocks. For example, you take maize. We have a lot of inbred lines. We know their pedigree history, so that we can able to cross based on their pedigree record. We can able to get an hybrid. But in case of some crops like mung bean, you take mung bean, no hybrids are there. But some unpreferred crops you can take. There we don't know they don't have some expensive pedigree records. So that case this may be helpful. Okay, we can able to propagate some superior heterozygous plants, even though we don't know anything about the genomics, anything we don't know. We can able to reconstruct it and we can able by simple cytological techniques without knowing any the reference sequence or anything, we can able to create, okay, propagate. Then we cannot use breeding at single chromosome level. How this possible? We can observe this. For example, in one line, parent one, all are having same background, but this having the different chromosome. Means the genes which are this chromosome is only separate. Then it the based on the phenotype, okay, all chromosomes are there. For example, you compare another phenotype, all having black chromosomes. Then you can able to tell the difference between them. For example, is a red color flower, it's white color flower, this plant. The chromosome, take this one only. It is a red color and it's a white color. So that if you do reverse breeding, there will be no recombination. Then the definitely chance of having red color genes in this chromosome is high. Is this correct or not? Very nice here. For example, take this is producing white and this plant producing red color. Okay. Then you can able to see rest of the other background is similar between them, but only this particular chromosome is different. And that's why we are getting phenotype is also different. Means the probability of the gene present on this chromosome is very high. So that we can able to study, we transfer the single chromosome in the different background. Then you can study what are all the genes which are present in this chromosome, means we can able to breed at chromosome level. We can able to study the trait at, okay, we can able to say that, okay, my trait is only on this chromosome. This is what the chromosome based chromosome breeding at chromosome level. Understood what is breeding at chromosome level?
Yes, sir.
Yeah. Then we can use marker assisted breeding, so that we can, with the use of markers, you can easily select which are the plants which having the proper the genotypic constitution, so that we can able to select easily, and we can able to cross, and we can able to use quickly. Okay, the time will be reduced as we discussed in marker assisted selection, and we can able to study several like profiling techniques like we can use for transcriptome metabolo, because everything at single chromosome level. The responsible having an haploid genome and single chromosome level. At chromosome level you can able to study the expression level of that particular gene, metabolism level of the particular gene. There are several examples. Okay, and you can able to what is the role of some genes majorly involved in heterosis, and we can able to study the heterosis effect on which gene, where they are present, by using integrating marker, double haploid, and reverse breeding. Three techniques. Okay. Then this is about reverse breeding. Okay, it's a very simple topic. No need to remember all the things. We will solve some questions so that you will get an idea how questions can be raised. See, this is in, I think this is 2020. The question was asked about reverse breeding. It's a breeding tool that can able to generate homozygous parental lines from a hybrid. This is true or correct?
True, sir.
Promotes the recombination in a heterozygote by knockdown of meiosis. It is true or correct? Sorry, true or wrong? True or false?
False, false.
False. Because it suppresses the recombination. Then a tool which uses in engineered meiosis. Definitely, we use knockdown or knock overexpress the genes which are responsible, which suppress the recombination. Okay, we are using an engineered meiosis. That's why it is also true. Uses the spore culture to generate the homozygous double haploids. We are doubling the chromosome, we can developing the homozygous intact chromosomes. It is correct or wrong?
True.
D. D. True. Okay. And aim to select the gametes having non-recombinant chromosomes. It is correct or not?
We are only eliminating crossover, sir, but segregation can
Aim to
happen
select the
also.
gametes having non-recombinant chromosomes. Correct or not?
True, true.
True. Because we don't want recombinants. Okay. That's why you can able you just an answer. B is wrong. Just directly cancel all the option. A, C, D, E will be your answer. Okay. Understood? So very questions will be very simple because there is the only review paper it is read by everyone. What are the things which are there in that review paper? They will definitely ask. Okay. That only can be asked. See, reverse breeding is a novel breeding tool which used to generate the homozygous parental lines from hybrid plant through engineered meiosis. Exogenous application of compounds that like mirin, which enhances the meiotic recombination. The statement, can you answer this?
Both A and R are correct. R is not the correct explanation.
How? Oh, I
So,
told. Mirin
A,
enhances, suppresses.
sorry, it suppresses. All
Suppresses. Okay. Suppresses the recombination. That's why it's we know the concepts. While reading in the paper, we'll do silly mistakes. At least I'll bet you, if you're not attentive in the exam, how much you read, how much your hard work, it is not depend. Like it's not matters in exam. How much you are attentive and how much you like your presence of mind is uh, the presence of mind up to 2 hours. The during 2 hours in the examination, it matters your rank. It decides your rank. Okay. Even see if you miss this, but you know the concept, but you miss this, means you're losing 5 marks, not 4 marks. Means you're attending this question by like subconsciously, without having your consent. You're apply. You're just with overconfidence. You're applying this option. You're opting this option. Means negative 1 marks plus 4 marks. 5 marks we are going to miss. 5 marks. The cost of 5 marks. Will rank will be decided from 1 to 10 rank. There is only difference of 4 marks sometimes. In my JRF, I think in JRF, like my marks is 480, 381, and my third rank is 382. What is the fourth rank is just 380. Fifth rank is 380 only, 379, 376, and three members got 376. See, not even one question. If you are in general, there are only 2 seats in IRA at that time for general, for MSc. See, if you are not getting, I was in general, so if I miss something, 1 marks also, I will be out of from IRA. This is the situation. You should be very, very careful while attending the questions. Then, a novel breeding tool which used to generate homozygous parental lines from a hybrid plant through engineered meiosis is called as reverse breeding, direct hit. Okay. What is forward breeding? We will discuss. We discussed already. What is forward breeding?
marker assistant.
Yeah. Means you are selecting, we are just avoiding all your self-food plants. We are just selecting true hybrids so that we are forwarding very fastly. There are several forms of forward breeding. This is only like you are forwarding based on the markers. This is one of the system. Okay. Then all you can know what is recombination breeding, mutation breeding is simple. Then they're all from different years. It's not from same year. You can by looking at the pattern of question, you can able to see this is one pattern, this is one pattern. They're all from different years. 4 years it is repeated continuously. A recombination by knockdown of meiotic crossing over, reverse breeding means it's correct. Recombination by knockdown of meiotic crossing over. Correct or not?
Also,
Recombination by knockdown, yeah. Suppressing the recombination, non-recombination by knockdown of meiotic crossing. Then selection of male and female spores having the combination of recombinantal parental chromosomes is also false. Non-recombinant should be. Then in vitro spore culture to generate the homozygous double haploid is correct. The selection of complementary parent to reconstruct this one. So that's why only C and D will be the right answer. Rest all other is wrong. Understood?
yes,
This is only simple. You read it this paper. Paper is also very in a simple language. Please read this paper. Okay. You just type reverse breeding review paper. You will get the paper. It is open access. Please read it and all the diagrams, information, slides, everything like the information bullet points, everything I taken from this paper only. And even all questions are directly copy pasted the lines. Just they change non-recombinant. They just remove the non. then they given the sentence like this. See, and all the questions are similar. They just made some changes and they are giving every year. Then another area where it's growing, it's like a transformative technology is speed breeding. Okay, rapid advancement of generation, RAG. Rapid advancement of the generation. Actually, normally we call TEK. Lee TEK is the father of speed breeding. It is not like a father, but he is actually first conceptualized and he used first time speed breeding in wheat. Okay. And this is a very important paper. You read it is also open access. It's a review paper, I think. Speed breeding is a powerful tool to accelerate crop research and breeding. Read this paper and we will discuss what is speed breeding. It means it's a rapid generation advancement achieved through controlled environmental growth chambers. Okay, these chambers meticulously control the light, temperature, humidity, CO2 levels, and we can give an ideal condition so that we can able to accelerate. For example, you take pigeon pea. One of my senior, actually, his name is Nunavat Ashwini. Fortunately, we lost her during floods. To develop 6 generations of pigeon pea in a single year. Pigeon pea is a 6 months crop. If you want to generate 6 generation, how much months you need? At least 36 months, means 3 years. But we can able to, if you use the speed breeding, she generated 6 generations of pigeon pea, and she used this sixth generation F6 as a real and use some identify the QTL. In 1 year, she developed the real population, identify the QTL, and multi-location trial of that one. Excellent research. Okay. This is the what the power of spreading. We can able to rapidly advance the generation in a single year. Okay, that's what called
So
rapid advancement of generations. What?
around the doubts.
Okay, okay, ask.
Can I ask tomorrow? The basic doubt.
What? What?
Basic doubt about this rapid advancement of generations. Whether
Yeah, you
this
can ask.
off-season cropping like wheat in would be SSD method in the
Yeah,
greenhouse.
that
They
also
also
rapid as well. Yeah, I have
they
slides.
those are considered as speed breeding or separately rapid advancement.
No, no, no. They are not considered as breed breeding actually, because we need
Okay.
some. We need to change. We need to look up the what is the photo period. Like we are not giving any ideal conditions there. Okay? But
Okay.
actually, now sometimes they use the terminology. If something is you are doing speedy in nature, that is speed breeding only. In Mungbin, our crop is only 60 days. Actually, I was giving my credit seminar on Mungbin. And actually, same Lihiki only as actually given on paper, building a better legume mung bean for the heat stress at reproductive stage. Like he only wrote on review paper. It also published in very good. And he mentioned all these advanced phenomics and speed breeding and all those things. Even I also presented the same. There was a question asked. Mung bean is just a 60 days. How much speed you want? Okay, I can able to take summer, spring, kharif, 3 crops in a year. If you go polyhouse, even I can take 3 crops. Okay, 60, like I have 60, 60, 60, 60, 60. So many crops I can take without doing all those things. That is also speed breeding. Some terminologies now you should go with the paper, like how the world is going on, you should move along with it. Okay? Means it's he defined speeding in such a way so that nowadays everyone is talking about this. But I think that question maybe not going to ask. Okay, giving the list and this is speed bidding or this. Don't think in that like. You understood the doubt?
Yes, sir. Yes.
Okay. We are using a controlled environment growth chambers to advance the generation. Why we need to control some? For example, you take long day plants. There is another question. Okay, long day plants. Wheat is a long day plant. If you give continuous light, means the photo period is length is very high. If you give continuous light, you can give an ideal condition. So the plant will accelerate all the phenological stages so that it will mature early, so that we can get the plant. If it is a total month is 6 months, within the 3 months you are going to get the complete maturity. That's why we're advancing. We are accelerating all the phenological stages by giving, for example, mung bean you take in a kharif, it will take 3 months sometimes. The same genotypes, and if you late sown, actually instead of I only, instead of kharif, sorry, instead of June, August, I sown in September 1. You know, both came harvesting at same time. The which are the crops sown in August, which are crops which sown in 1 month later. I harvested together all. Why it is happened? Because of there is a change in a photo period time. 1 month, August to September in Delhi, September it is a winter. The starting of winter, the photo period length, day length will be less, and our Mumbai is highly sensitive to photo period. Means if you change any sowing dates, the photo period will change and it will coincide with flowering. You may get drastic changes. The maturity, like the growth period, will definitely reduced if you change any growth period. It's not only reduced, sometimes it may like elaborate or shortens based on the nature of the photo period sensitivity of that crop. That where Lee Hickey controlled that photo period. He given optimum photo period length so that where we can accelerate the phenological stages. That is the crux. Okay. For example, and speed breeding. If you have developed speed breeding protocol for wheat, you can't apply the speed breeding same protocol for rice. Why?
Sir, the different crops need various photo periods and environmental conditions.
Why? Tell me in one word. Wheat and
Uh,
rice. Because where wheat is a long day plant and rice is a short day plant. I think. Am I right? Rice is
Yes,
a
sir.
short day plant or long day plant? Why you are thinking so much? What is the wheat is short day plant or long day plant? You're forgetting the basics means it's very difficult to crack any exam. Okay. Okay, you're
Sir,
thinking
wheat
so much
is
about
long
advanced
day.
basics. Yeah. Rice is short day. And most of the time winter crops are long day crops. Okay. Just try to remember these things. That's why we need a separate protocol. Okay, based on their photo period length as it is. That's why based on some short day plants, it's need another type of condition. Long day plants need another type of condition. And if you give drought, maturity will come fast in some crop. If you give water, some crops will mature earlier. There are different. That's why everywhere. I think you are getting. We are standardizing the protocol, speed breeding protocol for wheat. There will be paper on paper. If you standardize protocol for speed breeding protocol for pigeon pea, that is another paper. Means it's a new concept. Everyone started standardizing. Look at this particular condition. Okay, plant we are getting this early maturity. This particular condition like 12 hour photo period and continuous watering and this much hour of temperature, this much temperature. Okay, I'm getting this. This number of generation is created in a 1 year. It is continuously evolving. It is difficult. It is like environment to environment. It is different. Genotype to genotype. Some may be different. Species to species. It's different. That's why generalizing the speed breeding protocol it's not possible. Okay, based on the nature of the crop. Then how we can first phenotype. We can able to easily phenotype. For example, you want to develop a reel. What we can do? You have to wait 6 years. Instead, if you apply speed bidding, you can able to develop a real population in 1 year, so that you do QTL mapping in 1 year. In MSc also you can do QTL in 1 year. Okay, you can do QTL mapping in MSc also. Then you can able to study the phenotyping very. Then you can able to study the mutations. You can able to study the phenotype, the transformants. For example, transgenic. You are developing a transgenic, and you want to develop. You have to regenerate the plant. Then you had to generate M1, M2, M3, so that in M3 you are getting homozygous plant. Then it is fixed, transgene is fixed. Then say how much time it will require. For example, you take 6 months crop, 6 months duration. How much crop? See, creating transgene and validating and phenotyping, it will take nearly 10 years. You need a 3 PhDs for that. If you have a spirit breeding technique, you rapidly regenerate the plant, you rapidly create all the population. If phenotype within a 1 year, where all transgene validation and release will be possible. That's what the power of the speed breeding. Okay, understood what is speed breeding and what is the advantage of speed breeding? What is the
Yeah.
limitation also? I told limitation also. You can't generalize the protocol for all the crops. It's the greatest limitation. Then there are different speeding. You can just read. Okay, controlled environment chamber speed breeding or homemade growth room designed for low cost speed breeding. Okay, there are several ways. You just read it. Then we can able to do high throughput genotyping, genome editing, genome selection. We can integrate speed breeding with genomic selection, speed breeding with QTL mapping, speed breeding with MABB, speed breeding with genomic selection, or speed breeding with reverse breeding. There are so many review papers. They just make these things and they will publish the review papers. Okay, you just read all things. Then another term called as shuttle breeding. Okay. Then maybe in ARS always there is a question. What is the difference between speed breeding and shuttle breeding? We should be or there is what are the different techniques available to rapid advancement of generations? Speed breeding, shuttle breeding, off-season phenotyping. There are several things. Okay. You can use multi like the sheds, the polyhouses to create artificial conditions. Okay. What is shuttle breeding? Who discovered it? Anybody have an idea?
They have done first in Summit, Mexico. Noted.
Yeah, Norman V. Borland. Okay, in Summit. He actually conceptualized the shuttle breeding. It means that you are growing your generation in a contrasting environment to achieve 2 generations per year. Okay, it is the not only aim to reduce the generation time, but also another thing is in a contrasting environment location. What is the advantage? If you
So we
grow the
can
genome,
breed
if you
for
evaluate,
adaptability.
yeah, we can improve the adaptability. This is the greatest advantage of shuttle breeding. In IRA in rice, we have in wheat also, we have shuttle breeding program. Like in rice, Delhi one season and Adutharai in Tamil Nadu one season. The PhD had to grow 6 crops, 3 years, 6 seasons. One is in Delhi, one is in Adutharai. Okay, in rice, in wheat,
Wellington, Ooty.
yeah, Wellington and Delhi. And there is another called as some, I think in near to Arunachal, Himachal Pradesh. There is a one station is called as. There are 3 off-season trials will going to take place. Wait. Okay. Then means we can use constructing locations to achieve 2 generations per year. See, from North India to South India, there is so much difference. Okay. Now rice is growing in Adutharai, but it is harsh winter. We can't grow rice here. See the contrasting environment, so that we can able to develop the lines for the adaptability traits also. And for example, if there is a disease in one location, in Kharif season, but the same disease available in the other location in Rabi season, you can use this also. Means multi years, like in a single within without losing a season, In a single year, we can able to screen the genotypes for the disease rest. Even though in a single location, you will not find any inoculum. That is another advantage. Means faster breeding cycle, enhanced adaptability is a great one of the advantage of shuttle breeding. Okay, it is developed by Norman Borline at the International Maize and Wheat Improvement Center called SMH in 1940. Means, and it is successfully helps in the developing the photoperiod insensitive varieties and a wider disease resistance. Means, for example, in this location, they are having particular race of pathogen. In other location, they are having another race of the pathogen. In another location, there may be another race. So you are developing the varieties for all the races. This is also possible. N number of opportunities are possible due to this shuttle breeding. Okay, there was again the series of questions were asked in the ARS or any ASRB. These are the only things you have to read. What is shuttle breeding? What is speed breeding? And what is reverse breeding? These are the things very much important. Then, in comparison to speed breeding, what is the difference between shuttle breeding and this one? Shuttle breeding mainly prioritizes the achieving the broader adaptability. But speed breeding is just acceleration of breeding cycle. I think you got an answer for this, your question. Okay.
Yes.
Then, yeah, in what we are growing a chamber, just advancing the generation is the only our one objective. But here, the adaptability, opportunity of developing a line for different races, there are a number of opportunity other than the rapid advancement. Here we can take shuttle breeding utilizes naturally contrasting environment where your speed breeding relies on proper controlled chambers. Like you have to maintain the photo period, you have to maintain the temperature, you have to maintain the humidity, because all these are factors very much important for the growth and development of the plant. If we alter this, there will be change in the phenology of the particular plant. Phenology is nothing but flowering dates. Okay? Flowering date, when it will have juvenile flight, when will have reproductive phase, when will growth to maturity. All are phenological stages. Those are many are already working on it. Okay. This is like what is the contrasting nature of speed breeding and the this one. Okay. I hope you understood what is reverse breeding, speed breeding, and shuttle breeding. Why I'm getting giving a very comprehensive lecture means there should be a difference. Okay, when you are reading the book and listening the class, there should be a difference. Okay, like if I keep all the things and all the information and I will tell all the information by just reading the slides, what is the difference between the reading the book and listening the class? Okay, you should get an idea what are the things are there, how we can understand the concept, and you should able to think analytically so that you can understand better when you are reading the concepts. This is the main motto of my classes. You're happy with this? Okay, and please have any doubts, any queries, please text me or you can ask now. Any doubts? Stopping here. Understood?
Yes.
Yeah, okay. Thank you. If you have any doubts regarding any previous classes, you can ask. And actually, I'm thinking to start that every day I'm telling, but I'm not able to start generation of sequencing and advanced. Actually, I'm thinking to have at the end of course because I had to cover so like I will finish first the basic things so that the people will get interested because I'm continuously teaching the most difficult topics like mapping, detail mapping, reverse. Anybody has losing the interest actually. I think the live attendance for the live class or I know because it's some the habit. Like if you don't understand it, we are not able to like we're not trying to understand the concepts. We just avoid it. Okay, that's why I will finish the like heterotic breeding from tomorrow. I'll take heterotic breeding. It will take another 2 3 classes. So that I will finish heterosis breeding. Then I will start think quantitative genetics. Then I will come to your all omics based methods. Okay.
Okay. Yeah. Once you told your friend will take to one class about association machine. Whether it will be possible.
What?
Once you told that your friend sir will take
Yeah, that will
association
be
machine.
arranged actually. That's why I didn't thought some practical things about GBoss. What is Manhattan plots? What is QQ plots? And
Yes,
how we
sir.
can visualize LD blocks. That things I didn't thought because it will confuse you. And yeah, I will talk. Actually, I will take, but by looking at the attendance, I'm not able to call anyone. Okay, because the people are attending only very less like classes, even they have more students. But if you are because if we come with enthusiasm, at least you should have some people to teach. Okay, for me it's okay because I know that because I'm getting the analytics of those who are watching recordings, how much hour they're spending. It's a good actually. Many people are watching recording. It's not like that they are not interested. Many people are watching recording. They are asking the doubts. Okay, but if I ask someone means at least they should have some persons to discuss. Okay, some attendance is needed. Okay, that's fine. He's ready because he's beside my room only. That's why I'm not reluctant to ask anyone because so many are ready to take the classes. Even some who are past ASRB exam because I'm very close with many seniors and this thing. Okay. But I'm only not calling them because seeing the attendance. Okay. If you are the people are attending more live classes, like then I will definitely call. If no students were there, the attendance was less. So I have so many students. I didn't expect this many students. But that live class attendance is very low. That's why I'm not able to call anyone. Okay? If one day I will decide, like, if everyone is okay, like at least 80% of the strength is okay for live class, then I will call some guest lectures. Okay?
Okay.
Bye bye. Bye, good night.
Good night, sir. Thank you.